Good morning, good afternoon, and good evening, everyone, from wherever you're joining us, and welcome to today's webinar titled Mimicking the Ventricular Pressure Volume Relationship ex vivo with Cardiac Slices, featuring Dr. Bradley Palmer, CEO of Sarcometrics and Assistant Professor of Molecular Physiology and Biophysics at the University of Vermont. Today, he'll present to us the cardiac slice model for examining myocardial function, and he'll showcase techniques and results from a cardiomyopathy mouse model. I want to take this moment to thank our partners at scientist.com and the sponsor for this event, Ionoptics, for making it all possible. Let's welcome Dr. Palmer on. Good morning, Brad. Take it away.
Thank you very much, Sydney. Welcome. My name is Brad Palmer, and I'm a faculty member in the Department of Molecular Physiology and Biophysics at the University of Vermont. I am also founder and an employee of a preclinical contract research organization called Sarcometrics. The outline of this webinar will go as follows. First, we'll introduce the cardiac slice as an experimental model system of heart function, and we'll then demonstrate the preparation of cardiac slices from rat heart. And we'll walk through three functional measurements that can be made with the cardiac slice: intracellular calcium regulation, isometric force production, and mechanical work loops. And we'll also demonstrate detection of the Frank-Starling response, diastolic dysfunction, and a dose response to mavacamten in the cardiac slice. So this slide provides a visual summary of how we prepare and examine a cardiac slice. First, the heart is removed from the animal. The septum of the ventricle is cut longitudinally from base to apex and glued to a flat surface. And a razor blade cuts thin slices which are glued to clips. And the clips are then placed between a force transducer and a length motor for examination of mechanical function. The details of these steps will be presented a little later on in this presentation. And while mechanical function is our focus here, I'd like to point out a few other uses of the cardiac slice before we get going. For example, because the cardiac slice maintains the in vivo structure of neighboring cardiomyocytes, action potential propagation and other signaling between myocytes can be examined. Gene and protein expression can also be examined, as well as biochemical processes and post-translational modifications. Imaging of intracellular and extracellular structures and processes are also possible with the cardiac slice. Remember, the slice maintains the in vivo architecture of cells and extracellular matrix, while also being only a few cardiac cells thick. Therefore, preparing a cardiac slice is advantageous for many imaging applications. And long-term incubation is an important use of the cardiac slice. There are some laboratories that have been able to maintain functionally viable cardiac slices for weeks and even months, which allows for examination of cardiac remodeling after an intervention. The cardiac slice can also be loaded with fluorescent dyes that allows for imaging of intracellular calcium or action potential. And the cardiac slice can be attached to the force transducer and in that way allow for contraction relaxation function to be measured. If the slice is then also attached to a length motor, a force length work loop can be generated to mimic the pressure volume relationship of the ventricle. And so for much of this webinar, we will be focused on these mechanics of the cardiac slice. But what I would like to do is demonstrate how to prepare a cardiac slice from a rat heart. The first step is removal of the right ventricle. This still image shows the base of the heart after the atria and the valves have been removed. And the opening of the right ventricle is crescent-shaped, while the opening of the left ventricle is the round shape. And I'm going to show you a little video here that will last about 1 minute. In this video, the technician is grabbing the free wall of the right ventricle and trimming away that free wall to expose the septum. As the septum is exposed there, a blade, one of the blades of the scissors, can be placed inside the left ventricle, inside the LV chamber, and then a cut can be made from base all the way down to the apex of the heart so that the heart can be splayed open, which is an important part of the slicing process. So after the right ventricle has been trimmed away and the septum cut, the left ventricle is then placed on a vibratome for slicing. The still image that I'm showing you here shows the razor blade about to cut through the left ventricle. So now I'll show you a video on how this proceeds. So we're starting now with that ventricle that we had just prepared in the previous video. And we're going to put that ventricle onto a solid surface, in this case, a piece of glass. And that glass is, positioned next to a razor blade that will cut the left ventricle. The first item is to get glue. This is cyanoacrylate glue placed onto the flat surface. And now the left ventricle can be laid down epicardium side down, and it will just adhere to the flat surface. And it will only take about 10 to 20 seconds for the epicardium to adhere. And then we place a dissection solution, which is basically a Krebs Ringer solution with 30 millimolar BDM to prevent contraction. And we like to keep this solution cold, so you'll also see that in a minute, you'll see that ice will be placed around the bath here. And we like to oxygenate the solution during this whole process. Now I'm going to speed up the video so that you can see how fast you can see the process without having to wait for an entire slice. The frequency of vibration is about 80 hertz, and the amplitude of vibration is 2 millimeters peak to peak. And normally the blade speed is about 100 microns per second. And you might have noticed there that the bath was raised about 300 microns to allow for the slice to be 300 microns thick. This is a more modern looking vibratome, which is preferable if you have the ability to get a modern vibratome. This one, I think, is made by Camden. Oftentimes, if you set your blade range properly, the slice will just fall right off of the end of the heart. But as you can see in some of these examples, we had to cut the slice away from the heart. So one question that you may have is whether or not the slicing process destroys the cardiac myocytes. So several laboratories, including our own, have examined the viability of the intact cardiac myocytes within the cardiac slice. The left panel in this slide shows green color for intact cells, which dominate the image here. The red dots in this image represent strands of DNA that have been liberated during the slicing process. And the two arrows that I'm showing you right here at the top of the image point to the absence of either color, which indicates an intact nucleus of a viable cardiac myocyte. So if a cell had been lysed or disrupted during the slicing process, the nuclei would be shown in stark red, like the image over here on the right. Now this image, all the cells have been lysed by detergent to provide this negative control image. And we and others have found that the fraction of viable myocytes in a cardiac slice 300 microns thick to be about 90 to 95%. So now that we have produced cardiac slices, they must now be clipped. And so you see in this still photograph 2 examples of clipped slices that allow for functional measurements of calcium dynamics and contractile function. So now I'll show you a video of how we create these clips. This video is about 2 minutes long. We choose acrylic clips because acrylic doesn't have autofluorescence. Whoops, I'm going to back up here just a second. We start out with a cardiac slice that has not yet been clipped. And the first order of business is to visualize the directionality of the fibers and the directionality of the force production. And what you'll see here is a microscope view of that slice. I would like to point out that on some of the older vibratomes, you will get some sort of scarring, which shows the vertical lines that you see here in this slice. But the fiber dimension, or the fiber direction, the fiber direction is going left to right. So you want to make sure that you don't get fooled by any scarring from the razor blade. Once you've got the directionality of the fiber orientation, you can just use a razor blade on a glass surface to cut away the slice. We cut into a rectangular shape, and then again with cyanoacrylate glue, just simply glue the clips to the slice like that. And that's what your slice looks like at the end. So one of the last things you have to do as part of the preparation is determine the starting length of your cardiac slice. In the image on the left, you can see the objective of the inverted microscope. There's an objective right here just underneath the slice. With this inverted microscope, sarcomeres can be seen when the slice is less than 250 microns thick. You see in the example of sarcomeres on the right side. In this example, the sarcomere length has been set to 2.2 microns. We also tend to use, at least in our experience, even though you can see sarcomeres at thicknesses thinner than 250 microns, we tend to go down to 120 microns just to be sure of seeing the sarcomeres throughout the entire slice. If the inverted microscope allows for fluorescent imaging, then the slice can be loaded with a fluorescent dye, such as Fura2, Fluor4, or Cal-Red. In this example, a blue light is directed to the slice for the excitation of the dye. And the emitted light is depicted as green. And this green light can then be directed toward a detector. And then that way you can get a measure of your fluorescence intensity. A red light or some other wavelength that is not the excitation or emission wavelength, can be directed to the camera that is passed through the sample and passed straight through to a camera that allows for simultaneous monitoring of sarcomere length. Many of you are probably already familiar with recording intracellular calcium in isolated cardiac myocytes. In the example on the right, a single cardiac myocyte has been loaded with a fluorescent dye that is sensitive to intracellular calcium, and you can see the brightness with every contraction. When the calcium is high, in this example, the light intensity is high, and the calcium dynamics are represented by the fluorescent light intensity. For ratiometric dyes, such as Fura2, as I'm showing down here in this example, it is the ratio of the numerator and denominator signals that is proportional to the intracellular calcium. For the cardiac slice, however, there is an issue of motion artifact to contend with. Motion artifact arises when the sample moves in and out of the field of view, which occurs during muscle contraction. And this is to be expected for cardiac slices. If we use a ratiometric dye like Fura2 or CalRed, the motion artifact can be minimized. In this example, both the numerator and denominator show motion artifact, which is highlighted in the circles. But when the ratio is calculated, the motion artifact, which is mostly nullified, as you can see down here in the third trace of ratio. And it's often clean enough that you can get good characteristics or reasonable physiologically significant characteristics of calcium transients. But so far, our best results for measuring intracellular calcium have come from using CalRed. Following the lead of Sun et al., who published a paper earlier this year, the CalRed numerator and denominator are very bright compared to any motion artifact. And the calculated ratio, which is proportional to the calcium concentration, appears clear of any significant motion artifact. And so we have been using CalRed in all of our slices. So we can also record contractile force dynamics. And the cardiac slice, if the cardiac slice is attached to a force transducer, which is what we have here on the left-hand side of this image. Many of you in the audience are probably familiar with measuring contraction of isolated cardiac myocytes. In that case, the shortening and re-lengthening of the sarcomere length is recorded as a measure of contraction and relaxation function. Often the maximum contraction of sarcomere length is used as a measure of contractility. With the cardiac slice, the force transducer records isometric contractile force, and contractility could be taken as the maximum force per cross-sectional area of the slice. Although the shortening transient and the force transient are not strictly comparable, both can be used to quantify contraction relaxation characteristics. The cardiac slice permits detection of contractions simultaneously with the diastolic sarcomere length. I'm going to show you a video where you'll see that the force transients generated by the slice are recorded simultaneously with the sarcomere length. In the little video window, I'm sorry, I'm going to pause this. In the little video window down here, in this video window, you should be able to see the vertical running of the sarcomeres. And with each contraction, the sarcomeres are no longer distinguishable. But in diastole, the sarcomeres are clear and still enough that the detection algorithm can produce a mean sarcomere length, indicated by the red line. That is the peak of this red line down here. That indicates the mean sarcomere length. And in this example, the mean diastolic sarcomere length was 2.2 microns. So there is one more functional assay I'd like to present today, and that's the force length work loop. The cardiac cycle can be mimicked with the cardiac slice by controlling the length of the slice during its contraction relaxation. This can be done using a length motor indicated in this slide by the double-headed arrow on the right-hand side of the image. Specifically, if the motor shortens the muscle during systole and lengthens the muscle during diastole, we can mimic the cardiac cycle. But before we begin the demonstration, I'd like to take a closer look at the cardiac cycle so that everyone's familiar with the nomenclature. What you see on the left-hand side of this slide is the left ventricular chamber imaged by functional MRI. The light coloring indicates blood volume. And on the right side, you see the Wiggers diagram that displays the left ventricular pressure and volume relationship throughout the cardiac cycle. This audience is probably familiar with this relationship, but I'd like to point out 4 phases of the cardiac cycle. That is, when contraction begins, the left ventricular pressure is too low to open the aortic valve, and the pressure in the chamber rises without a change in volume. And this is called the isovolumic contraction phase, or IVC. And when the left ventricular pressure reaches the pressure of the aortic valve opens and blood can be ejected. And this is the ejection phase. And when the left ventricular pressure drops below the aortic pressure, the aortic valve closes and the myocardium relaxes without a change in volume. This is the isovolumic relaxation phase, or IVR. And then finally, as the left ventricular regains its original shape and relengthens, there is a suction developed, and blood moves from the left atrium to the left ventricle. This is the filling phase, which also includes an atrial kick in late diastole. If we plot the pressure against the volume, this pressure-volume relationship shows a cyclical loop. It's often referred to as a work loop because the area within this loop indicates the mechanical work generated by the pumping action of the ventricle. You can see the 4 phases of the cycle we noted in the previous slide. There's an isovolumic contraction on the right-hand side of the loop, shows a rise in pressure with no change in volume, followed by the ejection phase at the top of the loop, where pressure stays high and volume is reduced, followed by the isovolumic relaxation phase on the left-hand side, showing a drop in pressure with no change in volume, and followed by filling on the bottom of the loop, where volume is recovered at low pressure. Presenting the pressure-volume relationship as a work loop like this is useful for demonstrating the effectiveness or ineffectiveness of the heart as a pump. For example, compared to the normal work loop shown in red, the work loop with diastolic heart failure, shown in black, often demonstrates an elevated diastolic pressure during the filling phase. This is because the higher LV pressure is required to fill the left ventricular chamber. Because the change in volume relative to end diastolic volume looks relatively normal, this diastolic heart failure is often termed heart failure with preserved ejection fraction, or HFpEF. The pressure-volume relationship is useful for describing and evaluating pump function. And for that reason, we are trying to mimic it with the cardiac slice. So the cardiac slice can be used to mimic the pressure-volume relationship. If you look at the left-hand image, you'll see a cardiac slice that is already placed between a force transducer and a length motor. The contractile force, which is shown in the top right trace, is continuously monitored. And the motor position, which is shown on the second trace on the right, is controlled to assure that the muscle shortens during force production and then re-lengthen as the muscle relaxes. And the force feedback control is used to control the motor position relative to two force levels, an afterload and a preload. The afterload is specified as the force to be maintained or exceeded during the ejection phase, and the preload is specified as the force to be maintained during filling phase. The length motor is then controlled to re-lengthen the muscle to maintain this preload. As you can see in the video, the muscle is re-lengthened to get ready for the next cycle. In this slide, you can see the results of two different afterloads. The higher afterload, that is this higher afterload, corresponds to less extent of shortening. So you can see the shortening control to maintain the afterload will end up having less shortening if the afterload is higher. We can also control preload by changing sarcomere length. In this example, there are two sarcomere lengths that are being shown. We can expect different force production. In this example, the increase in sarcomere length results in an increase in developed force. And the blue traces are recorded after stretching the muscle 1%, and after the preload and afterload were adjusted to correspond to this to the new diastolic and systolic function characteristics of the muscle. The resultant work loops are shown here. Again, the orange traces correspond to the 2.2 micron sarcomere length, and the blue traces correspond to the 1% increase in muscle length. The slope of the end diastolic position can be used to measure the diastolic stiffness, sometimes called the diastolic elastance. This is noted on the slide as the EDFLR, or end diastolic force length relationship. And the slope of the end systolic position can be used as a measure of inherent contractility, sometimes called the systolic elastance. This is noted here as the ESFLR, end systolic force length relationship. So slope can be used to quantify the increase in contractility that occurs with increasing preload, also called the Frank-Starling response. And with this methodology, the use of the cardiac slice, myocardial performance throughout a cardiac cycle can be examined without having to fully instrument an animal or isolated heart. The slice offers control of sarcomere length and other conditions that are not possible with the other experimental model systems. So many of the previous examples that I showed you showed a mostly square work loop. And if we increase the afterload throughout the ejection phase, which is what happens in vivo, then we can generate a more physiologically looking work loop. In this example, the afterload starts low and increases over time. And in this way, the peak force ends at end systole, and we get a force length relationship that looks more physiological. Note that I've plotted the force length relationship here as stress versus sarcomere length, which allows me to better compare between slices of different dimensions, because the stress now is normalized to cross-sectional area, and the sarcomere length just indicates the actual sarcomere length that was measured in the cardiac slice. This slide shows a comparison of the Frank-Starling response in wild-type mouse versus a mouse with dilated cardiomyopathy. This DCM model is due to an absence of myosin-binding protein C. You can see in the wild-type response an increase in end systolic stress and with an increasing total mechanical work with increasing muscle length. But the cardiac slice with DCM does not show this increase in contractility with increasing sarcomere length. The DCM example also shows an elevated end diastolic slope indicative of diastolic dysfunction due to a stiff myocardium. In this particular case, we already know that the myocardium is fibrotic, which would explain the stiffness. And finally, I'd like to show a dose response to mavacamten detected in these cardiac slices. In this set of results, I'm plotting the developed stress against mavacamten concentration. And we see that with about 20 nanomolar mavacamten, we observe a 40% decrease in developed stress. And with 500 nanomolar concentration, we observe more than 90% reduction in developed stress. This result simply illustrates that the usefulness of the cardiac slice as a physiologically relevant model system for detecting cardiac responsiveness to drugs and drug candidates. So let me summarize. The cardiac slice maintains a syncytium of cardiac myocytes and fibroblasts found in vivo, thus maintaining the endogenous architecture and signaling between cells. And compared to isolated myocytes, experimental results from slices are more likely to have physiological relevance. Cardiac slices can be used to mimic the pressure-volume relationship of the whole heart while allowing continuous monitoring of sarcomere length in diastole. And with increasing sarcomere length, the cardiac slice demonstrates the Frank-Starling law of the heart, that is, the enhancement of contractility with increasing sarcomere length. And the cardiac slice model system is useful for determining dose response of cardiac muscle targeted drugs and drug candidates, and also useful for detecting cardiotoxicity. So I have a few acknowledgments. I would like to thank Matt Caparizzo at the University of Vermont for his collegiality and cooperation during the production of this webinar. I'd like to thank Fotis Batolis and Cesare Terracciano at Imperial College London for their workshops on cardiac slice preparation. And thanks to Sarcometrics and Ion Optics for sponsoring this webinar, and to Inside Scientific for hosting this webinar. And I'm going to leave with this slide up. This is a reference slide, so you can see the references that were for important papers. I think the Sun et al. Paper is especially important. The Patolis paper and the Watson papers, especially the very last one, the second to last one published in 2017. That's an especially important paper if you're just starting out. Thank you very much for your attention. I'd be happy to take your questions.
Thanks, Brad. Wonderful presentation. Let's get started with our first question. What is the advantage of generating work loops using cardiac slices versus catheterization and generation of PV loops in vivo with a PV catheter?
So one of the advantages of using the cardiac slice is that you can monitor sarcomere length, and so that you know exactly where on the force length relationship of the sarcomere you're operating. And you can also compare shorter sarcomere lengths to longer sarcomere lengths, because now you've got control over those sarcomere lengths. I mean, the PV loops are very, very valuable, but they can also be very difficult. For example, getting a PV loop from a mouse heart is very difficult. I think there's only about a handful of laboratories that can do it reliably using mouse heart, whereas the cardiac slice is quite easy to perform the work loop. We also, of course, have better control over the environment that the cardiac slice sees. Like, we can really specify the afterload and preload very quickly compared to a catheterized animal.
Excellent. I think that was very clear. Thank you so much. Our next question, could this be adapted for skeletal muscle slices?
Yeah, for sure. We've actually performed work loops using mouse soleus muscle. Now, even mouse soleus muscle is very fast, but we were able to produce work loops. I don't know that I would try an EDL, because that would be even faster. And we found that our motor has a frequency response of about 50 or 60 hertz, which is sufficient for most biological tissues. I mean, much more sufficient than necessary for most biological tissues, but for the very fast mouse skeletal muscle, it wouldn't be fast enough. But other than that, it would probably work just fine.
Okay, excellent. Thank you. Can you inject cardiomyocyte spheroids into a cardiac slice?
Well, that's a great question, in part because I don't, I had never considered that. I suspect that you can. If you're talking about something like iPS cells that can then infiltrate the cardiac slice, yes, that's happening. And what is happening is some laboratories are taking away the endogenous myocytes and replacing them with iPS cells that then differentiate into cardiac myocytes. So that'd be one way that you could use the cardiac slice, not so much as the cardiac slice per se, as a sort of substrate for the iPS cells. I hope that's relevant to your question.
Yeah, feel free to submit a follow-up if you have more questions off of that one, to the person who submitted. Thank you. Okay. We have another question actually from the same person. Can you dose a slice with endosomes?
Absolutely. I mean, um, you can provide just about sort of any sort of intervention you would expect to either inject into an animal or even just place into a culture of some sort. You can do that with a cardiac slice. It's funny. I when I was first working with our cardiac slices, I just thought it was going to be this massive piece of tissue that I really couldn't work with much. But I find that it behaves much like a single cell in many regards. For example, loading fluorescent dyes, its its response to intervention, it seems to respond as though it's a collection of single cells more so than a large piece of tissue. So I would think that your endosome question, I would think you would expect it to behave much like you would expect a collection of cells.
Okay, perfect. Thank you. Um, we have another question that says, thank you, Dr. Palmer. Seems like another benefit is to simultaneously image and quantify the calcium transients during acquisition of the work loops. Have you good experience correlating these parameters?
We've done it. We don't have a lot of experience with, and I'm so sorry I didn't present it because I could have presented that. Um,
Next time.
we yeah, maybe next time, exactly. And I don't have enough. I don't have enough data collected um, to try to correlate. But for sure, you can perform that analysis. We're already busy with the set of data that we've already collected without having done that yet. So we'll be collecting that sort of data coming up, and we'll be able to do those kinds of correlations. I just don't have any results to show you just now.
Okay, yeah, something to follow up on for sure. Um, the next question is asking if this has been tried with human atrial trabecular tissue or papillary muscles from larger species.
So for sure, the papillary muscles and trabeculae from larger species, whether it be rat or human um, or rabbit, that has all been done, and it works well. The atrial tissue, I'm just not familiar with. I don't know that anyone has done it with atrial tissue. I know that there is a group. I know of one group that is attempting it, and I suspect that they'll get results. I don't see a reason why it should not work with atrial tissue. I just don't. I'm just not familiar with the results.
Okay, yeah, we'll have to look into that further. Um, could you do you know whether we could inject virus for gene therapy to the 300 micrometer human slice?
That's a great question. I fully expect AAV to infiltrate the entire slice. However, I adenovirus, for example, will only infiltrate one, maybe two cells deep. And I don't have a good answer. Like, I can't guarantee you that your AAV will infiltrate the entire slice. It's just something you're just going to have to check.
Yeah, absolutely. Okay. We do have still quite a few questions, so yeah, please keep submitting them. This is a really great Q&A session. Thanks, Brad. Uh, is it possible to overexpress genetic encoded calcium indicators in slices? Um, there's a second part to this question. Is virus transduction your preferred method? And if so, how is the percentage of cells transduced? Let me know if I need to repeat any of that.
No, that's a great question. What we have done is we have injected AAV into the animal and then created slices, and we did, and we get excellent production of our protein of choice in that method, and it seems to be ubiquitously expressed. That is, every cell gets the virus, and we don't really have to be worried about heterogeneity of the delivery of the virus and the RNA. And in that particular example that I cited, we did have, it was a calcium, it was meant to deliver a calcium sensitive dye. It was a GCAMP. And so, yeah, we were able to deliver GCAMP to what looked like every cell in the heart. And then we generated the slices and, of course, measured the calcium transients from the GCAMP that was delivered from the virus. We did at one point try adenovirus in the incubator, and like I said, it didn't seem to work for us. And talking with other investigators, it seems even when they're successful, it only seems to infiltrate about one or two myocytes. So I'm getting the sense the adenovirus may not be the way to go, and the AAV would be the way to go. And like I said, the calcium indicator gets in there just fine. In fact, we're using that GCAMP as our reporter for the other proteins that we're also trying to deliver.
Okay. Yeah, great explanation. Thank you. Um, are the natural shortening speeds of the sarcomere and myocardial tissue in sync?
I'm so sorry. Could you repeat that question?
Yeah. Are the natural shortening speeds of the sarcomere and myocardial tissue in sync?
Oh, okay. I think that's kind of getting at a question of series compliance. If you're, if everything were, if the slice and the clips and the hooks and all of the machinery, if all of that apparatus were perfectly stiff, then you would expect the sarcomeres to be perfectly in sync with muscle length. But because there is some compliance, that is, the glue may be slightly compliant, the hooks, the clips may not be so stiff onto the hooks. Maybe there's a little bit of give in the connection between the clips and the hooks. Well, then the sarcomeres will contract more than the muscle. So you've got to be just aware of that. It's just a question of how much serious compliance you can tolerate. There's always going to be some level of serious compliance. We've measured it to be on the order of contributing only single digit error in some of our measurements. So below 5% error in the sarcomere length versus full muscle length. So we tend not to be overly concerned about it right now, but it is something you should be concerned about at the very beginning when you're just trying to figure out your initial conditions for your experiments.
Okay. If there's any follow-up to that, please feel free to submit. But I think, yeah, great job, Brad. I believe you did already answer this. Somebody's asking if this could be used for papillary muscle only instead of whole slice of the ventricle.
Yeah, we've done it with, like I said, we've done several studies with papillary muscle from rat. I would not recommend doing it for mouse. Mouse papillary muscles are very, very small. And if you try to prepare them, at least with the methodology that we've been using, you'll find it very frustrating. And so we got frustrated within just a couple of days and realized, the amount, the number of papillary muscles we would examine would be so small that we just didn't want to go through the motions. Whereas creating a cardiac slice from a mouse muscle, even though it's more difficult than a rat muscle, it's still quite fine. We can make 6 to 8 cardiac slices from mouse muscle, and so you've got now 6 to 8 samples from that mouse muscle. Whereas you might only have 1 or 2 samples of papillary muscle, and even then just be too frustrating to actually examine them.
Okay, makes sense. Yeah, that's there was about 34% of respondents who work with mouse cardiac muscle. So
Right.
for them to keep in mind, absolutely. Let's keep going. We do still have quite a few questions to get to. If you're good to go, get some water. Let me see what the next question is here. Okay, how many cardiomyocytes are captured for the estimation of the sarcomere length output? Or how is the readout signal distributed over the slice?
That's a really great question because there is undoubtedly some heterogeneity in the sarcomere length. And so, what we tend to do is we do tend to look around the slice, pick out 3 or 4 measurements, and kind of take an average of those 3 or 4. The variability is on the order of 5 to 10%, depending on the slice and a disease model, for example. Um, but we feel fairly confident if you're talking about a chain. We'll take an average of that 3 or 4 spots around the slice. But then we'll kind of settle as that is the sarcomere length for that muscle length. And so if we change the muscle length 1, 2, 3%, whatever that is, we can then assume that we're getting a similar sort of change in sarcomere length. That's the best we can do. I think you just have to recognize and tolerate that there is going to be some heterogeneity.
Mm-hmm. Okay. This one's a short question. Can you cryopreserve heart slices?
You mean to use later? I don't think so. And when I say, and I'm taking cryo to mean freeze them. And my understanding is if you were to freeze a cardiac slice, just like any other tissue, it would destroy the cell membrane. And if you destroy the cell membranes, that's really no longer viable. But but along those lines, we have taken these cardiac slices, and we usually just leave them on ice, and you can use them for hours, and we get very, very reasonable force production and relaxation function if they've been kept on ice in BDM for hours. We can even sometimes, you're at the end of the day, we've put them in the refrigerator and left them overnight in the refrigerator, and the next day they still will give you reasonable contractile performance. They will not be as good as fresh overnight, but it's very reasonable. So if you're practicing or if you're trying to develop a protocol, you can use slices from the day before. It's very handy for that.
Okay, excellent. Yeah, I recall seeing in some of your videos or slides that perhaps they were on ice originally. So thank you. For the mavacamten data, what are the loading conditions, preload and afterload?
So in that study, the preload was the force that corresponded to a sarcomere length of 2.0 microns. So we would stretch the sarcomeres, stretch the muscle until the sarcomere length was on average 2.0, and that would be the corresponding force, or the corresponding diastolic force in that case, would be our preload. And then the afterload would be 50, I believe it's 50% of the developed force in addition to that preload. So if the developed force was 10, the total developed force was maybe 10 uh, millinewtons, then we would set the afterload to 5 millinewtons above the preload.
Okay, excellent. Hopefully that helps answer the question. I think this next one will be the last question with the time that we have left. And hopefully you have some insight into this. So the question is, how can data obtained ex vivo be used to develop new drugs or medical devices? If you have any insight.
Well, there's two things that I'm going to answer in two ways. First of all, every drug, whether it be cardiac targeted or not, every drug has to demonstrate that it's not cardiotoxic. And so it's important that you have a model system, whether it be isolated cardiac myocytes, or in this case, the cardiac slice, where you can apply your drug or drug candidate until you get to the point where you notice that the drug is toxic. And this is every drug is going to be toxic at some point. So the dose makes the poison, and so that's one way that you can use these sorts of experimental model systems. The other way is you want to be able to demonstrate if your drug or drug candidate is meant to target cardiac function. Then, of course, you want to be able to demonstrate at what dose do you get a certain response, and do you get any other response that you were not hoping to find. Like if your drug candidate was meant to help relaxation function, but it actually does something to contractile function, well, then you want to be able to characterize that. And it's very important to demonstrate all of the responses that you can get, whether it be from a single cell, or in our case, I think the cardiac slice makes for a really good way of controlling all of the conditions of the experiment and controlling the dose, that is the concentration of your candidate, so that you have a really good characterization of what that drug actually does to the myocardium.
In closing, we hope you enjoyed this Inside Scientific webinar produced in partnership with Scientist.com and sponsored by Ionoptics, and I hope you all enjoy the rest of your day.